I'll use this lecture to discuss how matrix approach offers a unified diagnostic system for uncertainty analysis. I'm Ichiro, a professor from Cornell University. The unified diagnostic system is also called a 1-3-5 scheme of diagnostics. 1 stands for one formula to unify the land carbon cycle models, 3 for 3D space to evaluate all model outputs, 5 is for 5 traceable components to pinpoint uncertainty sources by traceability analysis. These slides show 2 forms of equation. The diagnostic form on the top and the dynamic form at the bottom of the metric equation. The 3 dimensions for diagnostics are response time, MPP, and the carbon storage potential. The 5 traceable variables are MPP, plant allocation coefficients, decomposition coefficients, carbon transport coefficients, and the environmental scalar. Now I review the background of why we need new method for uncertainty analysis. It is well known that the model prediction of a land carbon cycle is highly uncertain, as shown by this famous figure from Pierre Friedrichstein and his colleagues published in 2006. In response to climate change, predicted land carbon sinks are widely different among models. The uncertainties in model predictions are documented almost in all model intercomparison projects, MIPS. Uncertainties sometimes become larger instead of smaller as we incorporate more processes into models. We cannot easily identify sources of uncertainties to effectively address this issue. As Carlos Sierra and his colleagues has pointed out, increasing details in process representation in models and the simulations they produce hinders our understanding of a holistic system behavior. This is a so-called modeling conundrum. Their commentary paper suggested that the metric representation of land carbon cycle models provides a general framework for the qualitative understanding of models without compromising details in process representations. The unified diagnostic system offered by the metric approach is based on one formula to unify land carbon cycle models. In previous 2 training units, we have showed you that the metric equations can unify the land carbon cycle models. We have converted nearly 20 models to the metric equations. Those models include 4 versions for CLM, OCD, and CABLE. Carlos and Marcus Mueller from Max Planck Institute for Biogeochemistry reviews all soil carbon models according to the different numbers of principles included in models. The metric equation we have focused on in this training course satisfied 5 principles: mass balance, substrate dependence, heterogeneities of decomposition rates, transformation of organic matter, and environmental effects. When decomposition rates or transfer rates are functions of a substrate, we still can use a metric equation to describe carbon dynamic. In this case, the metric equation becomes a nonlinear model. Indeed, Carroll's group uses the nonlinear metric equations to represent more than 10 microbial models. That we can understand models at a different levels of complexity under one overarching theory. A question still remains: how does one formula unify all the land carbon cycle models? The differences among the models include different numbers of pools, different ways to connect models, using different forms, for example, a fixed value, a simple equation, or a nested model to represent a process, and different parameter values. The metric equation also allows new process to be incorporated into any models. For example, here are 8 models. Among them, CAM has 2 pools, and OCDE has 100 pools. Pools are connected in different ways among the 8 models. All the 8 models have been represented in the unified metric form, though. For a given process, such as the mortality rate of plant, can be represented by a fixed value in a model, by a simple equation of a plant functional type in another model, or by a nested submodel in a dynamic vegetation model. The different ways of representation lead to the different level of model complexity. Models are often different in assigning parameter values. For example, CABLE allocates 23% of photosynthesis to the wood, whereas CLM3.5 allocates 16% to wood. That difference allocation coefficient alone can lead to the differences in residence time. Now let us explore what the 3D space mean for evaluating model outputs. My lecture for Unit 1 on theoretical foundation of the land carbon cycle and the metric approach has explained that we need 3 terms: carbon input, residence time, and carbon storage potential to describe the transient dynamic of land carbon cycle. The transient dynamics also present in our papers published in 2017. The first term on the right side of the equation describes the steady-state carbon storage capacity. The first dimension of transient carbon dynamic is the carbon input through primary production. The primary production, including gross primary production GPP and net primary production NPP, quantifies the amount of carbon that enters an ecosystem to go through a variety of processes of land carbon cycling before released back to the atmosphere. The second dimension of transient carbon dynamic is the carbon residence time, such as those shown in these maps, which were estimated from data assimilation. The residence time quantifies the duration of carbon staying an ecosystem before carbon is released back to the atmosphere. The 2 terms, NPP and the residence time, together quantifies equilibrium carbon storage capacity. In these slides, we show simulated carbon storage capacity by 3 land models: Beijing Climate Center (BCC) model, Canada CIN model, and the Community Earth System Model Biogeochemical Modules CESM-BGC. In response to climate change, all the 3 models simulate increase in MPP but decreases in residence time. CESM-BGC simulates the smallest MPP and residence time, leading to the lowest carbon storage capacity. BCC model simulates the largest increase in MPP, leading to the largest increase in carbon storage capacity. The Canada model simulates the highest residence time that estimates the highest carbon storage capacity. However, when models simulate responses of land responses to climate change, carbon cycle is no longer at equilibrium. In this case, we need the third term, carbon storage potential, to describe disequilibrium of the land carbon cycle, which is represented by color in these slides. Let me use one more example to explain the third dimension of land carbon cycle. This photo shows free air CO2 enrichment phase experiment in Duke Forest in North Carolina, USA. The phase experiment started in the middle of the 1990s and ended in 2010. CO2 concentration in the treatment's rain were elevated by 200 parts per million. To illustrate the carbon dynamic in the phase experiment, let us make a few assumptions. Let's assume that MPP is 1,000 gram carbon per meter square per year, and the residence time is 40 years at ambient CO2. Then we have the steady-state carbon pool size in Duke Forest to be 40 kilogram carbon per meter square. We assume that elevated CO2 increased MPP by 40% but has no effect on residence time. The steady-state pool size is 56 kilogram carbon per meter square at elevated CO2. For the sake of simplicity, we also assumed no seasonal and diurnal changes in forcing variables and carbon processes. Now we have 2 equilibrium states of carbon storage, one at ambient CO2 as X subscript 0e, and the other at elevated CO2 as X subscript 1e. At the very beginning of a phase experiment, we need to determine which direction and how fast the carbon storage would change. Generally speaking, we expect that the carbon storage change towards the equilibrium state at elevated CO2, but the rate of change is faster in the first few years, slow in later years. Now we can see that current carbon storage X moves towards XE. The difference between equilibrium carbon storage and the current storage is the carbon storage potential X sub P. The rate of carbon storage change is proportional to the potential. Now we have learned 3 points about the carbon storage dynamic from above example. First, carbon storage is always moving towards the equilibrium capacity. Second, the capacity is the ultimate drive of carbon storage potential. Third, the rate of carbon storage change is proportional to the carbon storage potential. However, the equilibrium carbon storage capacity is changing over time. The equilibrium capacity equals MPP times residence time. Because both MPP and the residence time vary with time, the capacity also time dependent. This is a reason why carbon cycle in terrestrial ecosystem is a non-autonomous system. We simulated carbon storage dynamics in Havel Forest. This slide shows the seasonal change of fine root biomass. The equilibrium fine root carbon storage is the theoretical quantity, which is a function of MPP times residence time. As MPP is low in winter, high in the summer, the carbon storage capacity, as indicated by black line, is also low in winter and high in summer. The red line indicates the current carbon storage in fine root, which is higher than the black line in the winter, lower than the black line in the summer. The red line is always moving towards the black line. That is a mathematical explanation why the fine root declines in the fall and the winter but increases in spring and the summer. For a non-autonomous system with both MPP and the residence time are changing with time, the carbon storage capacity still controls the direction of carbon storage change. And then carbon storage potential still determines the rate of carbon storage change. Therefore, the carbon input, residence time, carbon storage potentials form a 3D space within which all model outputs can be evaluated. Sanzo has applied the 3D to evaluate model performance from three model intercomparison projects, 3 MIPS, CMIP 5, Trendy, and MIST-MIP. The 3 panels on left use MPP as x-axis, residence time as a y-axis, color for carbon storage potential to place outputs from 25 models in the same space to evaluate their performance. The 3 right panels use the smooth line to indicate the current carbon storage, the shaded area to indicate carbon storage potential, the zigzag lines to indicate carbon storage capacity. The right panels clearly show time courses of those quantities. This is the first time we are able to evaluate all model outputs from different MIPS in a simple 3D space. Enqing He has recently developed a matrix MIP with 8 matrix models. The 8 models vary greatly in number of pools and soil vertical layers. With the same carbon input and through MPP, Enqing has showed that the uncertainty among 8 matrix models expands in simulate the cumulative carbon production at the plant, soil, and ecosystem scales. The spreading has been observed in many model intercomparison projects. With the metric equation, we can examine the causes of uncertainty. Enqing standardized those traceable components, which are A, C, K, and B in the matrix equation one by one. The uncertainty among the eight models is gradually disappearing. This study demonstrated that matrix approach can empower research on model uncertainty. Now let's examine the 5 traceable components of a carbon cycle dynamic. The matrix equation describes carbon input via MPP, allocation of carbon input to the different plant parts, decomposition of organic matter, carbon transfer among pools, and the environmental scalar. They consist of 5 components of the matrix equation. Mathematically, the 5 components are largely independent from each other. Moreover, each component can be further traced to their subcomponents until individual carbon cycle processes and their parameter values. These mathematical property enables us to trace sources of model uncertainty to individual process and parameter. This traceability analysis was first developed by Jin and Xia for the equilibrium carbon storage capacity and expanded by Li Fenjiang and Zhou San to transient dynamic of land carbon cycle. This slide shows the traceable pathway from carbon storage hierarchically down to the vegetation traits, climate forcing, and soil attributes. These traceability frameworks enable uncertainty analysis to be analytically transparent. Here are examples of traceability analysis. Uncertainty of 9 models in CMIP5 on the up left panel can be attributable to variations in MPP for 53%, residence time for 46%, carbon storage potential for 1%. Then the variations in MPP among the 9 models can be further attributed to variations in baseline MPP for 94%, temperature scalar for 5%, and water scalar for 1%. Here is another example. Australian CABLE model predicts a lower carbon storage capacity than CLM CASA prime due to the lower MPP, but CABLE has a higher residence time than CLM CASA prime. The higher residence time in CABLE is mainly caused by setting lower decomposition coefficient, leading to a higher baseline residence time. The environmental scalars among the 2 models are similar. Those examples all show that the traceability framework can help trace sources of model uncertainty down to the individual process or parameter values. Enqing He led a research team to apply the transient traceability to a matrix model intercomparison project, so-called matrix MIP. The transient traceability analysis allows them to precisely attribute model uncertainties to the 6 components of the matrix models. They find that the baseline residence time contributes most to the uncertainty in the matrix MIP. This training unit will show you how we can add diagnostic variables in matrix models so that we can use the 1-3-5 scheme for uncertainty analysis and traceability analysis. Unit 5 will particularly show you how you can do traceability analysis.